Okay, today's lesson is about the structure of DNA, starting with first the monomer. The name of the monomer or building block to a DNA strand is called a nucleotide, which is composed of 3 parts. It's a 5-carbon sugar, a phosphate group, and a nitrogen base, sometimes called a nitrogenous base. Structurally, it looks just like this. A central sugar, so I'll correspond that to part number 1. On one side, what you get is the nitrogen base, and the nitrogen base comes in 2 varieties, which we'll talk about in a few seconds. And then we also have a phosphate group. So that's the second part, and that's the third part. And we named these bonds back a few chapters ago. This bond right here is the N-glycosidic bond. This bond is the phosphodiester. Nitrogen bases, as I just mentioned, come in 2 varieties. The purines, which stand or represent a 2-ring nitrogen base, and the pyrimidines, which are a 1-ring nitrogen base. So we'll draw them, but let's first talk about the different nitrogen bases. There are 4 of them in DNA. The purines are adenine and guanine, and what they would look like is that central sugar. I'm going to go over the thing, but here it's a double ring. So we have 2 rings of nitrogen bases with the phosphate group. Whereas the pyrimidines are cytosine and thymine, and they will look, there's your central sugar right there. The phosphate group is the same, but now there's only 1 ring for the nitrogen base. So two of the 3 parts are identical. The only difference is the nitrogen base, whether it's 2 rings or 1 ring. The base pairing rules. DNA is a double strand, so you often see it drawn. That's 1 strand, and then it looks as a helix. It kind of wraps around itself. So what will happen is the nitrogen bases will pair up with each other. The left strand will unite with the right strand via a bond, which we'll talk about eventually. But what you can do is, if that's the N-glycosidic bond, it connects to nitrogen base A. The base pairing rules say A pairs with T and G pairs with C. So if A is here, I can predict that T would be present on the other strand. Down here with a little bit more room, if C is on this strand, G must be on that strand. So if I am given the sequence of nitrogen bases that follow this line, I can predict what is the sequence of nitrogen bases on this line because of these base pairing rules. And these are sometimes referred to as Chargaff's rules. So that's the first rule, that these 2 pair just like that. The second rule, and this is written down below, I just was running out of room, so you can write that down in your notes below. Purines pair with the pyrimidines. Adenine being a purine, thymine being a pyrimidine, purine, pyrimidine. This is your purine paired with a pyrimidine. So that's just structurally another thing to take note of. So the real big part of this chapter is going to be drawing your DNA strands at somewhat of a detailed structure. So here I started drawing it. What you're going to do is take that box that's on the next page, and I want you to turn it so that it is more vertical rather than horizontal. It's just going to be easier for you to draw your first diagram just like this. So here's what we did. I started drawing, and you can draw in the upper left-hand corner, giving a little bit of space up here. I drew 1 nucleotide. So here is my 5-carbon sugar, here is my nitrogen base, and here is my phosphate. What I did not teach you yet, but you will now understand, is that every corner of this pentagon, which represents the 5-carbon sugar, represents a different carbon. And to signify which one you're looking at, you actually number them. The one that attaches to the nitrogen base is always labeled the first carbon, and then will work in a clockwise motion around. So the phosphate group is always attached to the 5th carbon. So that's very important when building the nucleotide, that you just don't sporadically throw the bond wherever you wish. You actually bond it to the correct carbon. So what I'd like you to do is draw the 4 more nucleotides. So what I'll ask you to do, this is the middle of the second one. I'm not going to do them. I'll hand draw them now. I'm going to alternate the different types of nitrogen bases, but notice what I'll do is I always draw off of the first carbon the nitrogen base, and off the fifth carbon the phosphate. So continue drawing. I'll probably work a little bit faster than you, so you actually might be able to pause this video. But here I have my fifth one. The order of the nitrogen bases, it does not matter. I'm sure that you're going to copy exactly what I drew here. But notice what I have here are 5 individually non-bonded nucleotides. So what I have to do actually to build a polymer of DNA, I have to bond that. So I have to connect this first nucleotide with the second one. And the quickest and easiest connection is actually going to be found right here. So I'm going to draw a line, and this line signifies actually dehydration synthesis. I'm not going to draw it. I can't actually see it happening. You just have to understand that what just happened there in that line was the removal of water. We have dehydrated a lot of molecules before. So what I do is I'm just going to keep on going through, always bonding the phosphate group to the third carbon. So in this case, it's that bottom left carbon. Now, I've dehydrated it 4 times, thus producing a 5 nucleotide polymer. So this is the left side strand of the molecule. And I can kind of go in here and just make up letters based on the structure. If this is a pyrimidine, this could be T. This is a purine, so it could be A. Another purine, it could be G, C, just to get all 4 types. And then we're back to potentially G to kind of match the shape of the nitrogen base. Let's also label the last one down here. It's kind of important, these end ones, that you represent the orientation of the carbons, because what we now are going to do is build the second half of the DNA, the right-hand strand. And because nitrogen bases, when 2 DNA strands join together, their nitrogen bases actually join together. So if I drew the right-hand side in the same orientation as my left-hand side, my nitrogen bases would not be facing. If you kind of follow the cursor, if you structurally drew, if you kind of do the shape that I'm doing, that's the nucleotide, my nitrogen base will be facing the same direction. So what I have to do is I'm actually going to flip this around. So when I do that, I actually have to flip the orientation of my sugar. So this is a little bit harder. You can actually flip your paper upside down if you'd wish. This might be easier. But now I'm going to draw my sugar. Notice my sugar doesn't, the point really is pointing down now. So this is my 5th carbon. It's actually pointing downward as opposed to pointing upward. So this is my 1st carbon, 2nd, 3rd, and 4th. So off of the 1st carbon, I now have my nitrogen base. Now it's facing the actual nitrogen base of my left side strand. And remember that if the left side strand has T on it, what letter must be this nitrogen base? Hopefully you're thinking or said A. Off of the 5th carbon comes my phosphate group. So here's the orientation of my right-hand side molecule. So what I'm going to have you do is continue drawing. These are what are known as complementary sequences. So I'm going to draw my sugars first to kind of give me a spacing of where I need to draw. Sorry, my hand drawing is not the best. I'm really exaggerating, I'm sure, the point with the 5 carbon, but it's really, really important. So each of these has the 5 carbon, so then off of the 5th carbon always you have the phosphate group. Sorry about that. Phosphate. Sorry, I'm running out of room. And then each of these is my 1st carbon, so we have our nitrogen bases. And if this is a purine, this must be a pyrimidine. This is a purine, this must be a pyrimidine. That was a purine. There's my pyrimidine, pyrimidine. And you can press pause if I'm working a little too quick with the drawing. The last thing I have to do is just connect. Once again, dehydrate. Dehydrate, I have to go backwards, sorry. So I basically connected now my right-hand side of my DNA model. The last thing I have to do is draw the connection. There actually has to be a bond between the left and right-hand side. Right now there's a space between them, because I want to draw what is a hydrogen bond. So I'm going to draw it as a dotted line because it's technically, we've learned about hydrogen bonds a lot. They're just an attraction between two polar atoms. But notice I drew two of them because every time you have an AT pairing, you actually have two hydrogen bonds. So I'm going to label this. This is not a double hydrogen bond. It's two individual hydrogen bonds. And if that's an A, this has to be a T, so that's also going to be the same number. Between G and C, you actually find that you get 3 hydrogen bonds. So you can draw a third dotted line to represent that third hydrogen bond, and we have them right in a row. So there is my full picture right there of a DNA. What I'm now going to do is just draw, again, on the bottom right, the numbers again, because it's really important. 1, 2, 3, 4, and 5. The last thing you have to understand or learn in this lesson is how they name the corners of a DNA strand. So here we have the 4 corners of the DNA. What you do is they label the ends of the DNA based on which carbon is most extreme or most facing outward. And they choose between the 3 and the 5 because they're basically on opposite sides of the sugar. So for example, this upper right-hand corner, the 3 carbon is the most extreme or outward carbon. So what they do is they call this the 3 prime. I don't question why, just they use the word prime for some reason, but they say this is the 3 prime end of this strand. On the alternate strand over here, look at what happens. We have the 5. So they call this the 5 prime end. Down here, what number is most extreme? But the 3 prime. And over here on the right, what number is furthest out? But the 5 prime. So what you get here is the idea that DNA from the left and right side do not expose the same side of the sugar. Also notice the orientation of the phosphate. On the left-hand side, or the 5 prime side, the phosphate is most extreme. On the 3 prime side, the phosphate is facing downward. That's another way to figure out which side is the 5 prime and 3 prime, which is a skill we have to develop, is understanding how to identify the 5 prime end and the 3 prime end. So you have to know how to number the carbons, and you have to know where the attachment points are for the other parts that attach to the sugar. So for right now, that's what we want to talk about, just the structure of DNA. I'll be around to answer questions, but I just want to show this as a little introduction so we can go and move forward the next time I see you.